Chapter 7. This video is going to provide you an overview of energy metabolism in an aerobic environment, that's with oxygen. We're going to cover what happens to glucose via glycolysis, the TCA cycle, and the electron transport chain. So this picture is directly from your textbook, and it is an extremely simplified overview of the energy yielding pathways. So in a hamburger, we have protein, we have carbohydrates, and we have fat. And this just shows you that proteins get broken down into amino acids, carbohydrates into glucose, and fat into glycerol and fatty acids. We're going to talk about right now, this pathway here for the carbohydrates that use glucose to turn into pyruvate, pyruvate into acetyl-CoA, acetyl-CoA goes into the TCA cycle, and the hydrogen ions that are produced from the cycle can be shuffled off to the electron transport chain to make more energy. So glucose can be converted into pyruvate, and we do that via glycolysis. So glucose, we're going to show you on the next slide, is a 6-carbon molecule that gets split into 2 3-carbon molecules. What I want you to know here is that pyruvate has 2 options. In the aerobic environment, in the presence of oxygen, it turns it to acetyl-CoA. In an anaerobic environment, which is the absence of oxygen, like your muscles when you're working out, it can turn pyruvate to lactate. And we're going to talk about this. This is going to be the Cori cycle. That'll be its own video. But this, what we're going to talk about first is pyruvate to acetyl-CoA. So glycolysis is the process that breaks glucose, a 6-carbon molecule, breaks it into 2 pyruvate molecules. And these are both 3 carbons each. And I wish it was just that easy that we just split the carbon in half and got 2 pyruvate molecules, but it's not that easy. This are all the steps, or this shows you all of the steps of glycolysis. We go from glucose to glucose 6-phosphate and down the line this way. And we end up, after these 10 reactions happen, we end up with pyruvate. So what I need you to know is that glucose gets turned into pyruvate. In this 10-step process, these are the 10 steps, and this occurs in the cytoplasm, right? Remember, we're talking about energy metabolism in the cell, and glycolysis happens in the cytoplasm. Everything else that we see from here on out is going to happen in the mitochondria. So, it occurs in the cytoplasm. It's a 10-step process, and it requires a little bit of energy to actually have this process occur. So, between glucose and glucose 6-phosphate, we have to add ATP to the mix in order for this reaction to happen. And then over here, we have to use ATP again. So we need 2 molecules of ATP to make glycolysis happen. And here we get some phosphate groups and an ATP and another phosphate group and more ATP. So we spend 2 ATP to make 4 ATP. And when we repay ourselves back these 2 ATP that we spent, glycolysis yields or nets us 2 ATP. So we just did this part of the process. We went from glucose to pyruvate. This little arrow here represents glycolysis, and that's that 10-step process that I just showed you. So now that we have all of this pyruvate, we have 2, for every molecule of glucose, you get 2 pyruvate molecules. What are we going to do with that? So here we are in the cytoplasm with our pyruvate, and now we're going to move into the mitochondria. And when we do that, we lose some CO2, carbon dioxide, and the remaining molecule picks up a coenzyme A, and this is the result, acetyl-CoA. Acetyl-CoA is important because this is what we can use in the TCA cycle. So now, from here on out, like I said, we're in the mitochondria. So we have pyruvate that gets converted to acetyl-CoA. It loses this carbon. It also releases some hydrogen ions, which we can use later on to create ATP. So now we've just gone from pyruvate to acetyl-CoA. Brings us to the Krebs cycle or the TCA cycle. I usually call it the TCA cycle. The Krebs cycle, it's the person who, I'm going to use the word discovered, but discovered or identified the Krebs cycle. It's named after him. There are some other names for it, like the citric acid cycle or the tricarboxylic acid cycle. I use the TCA cycle as probably the easiest one. So it's a cycle. So it starts and it ends kind of in the same place. And we're going to use those acetyl-CoAs that we just created to start the process. So what I want you to know here is that this oxaloacetate is a 4-carbon molecule. Acetyl-CoA is a 2-carbon molecule. When we put acetyl-CoA into the TCA cycle, it combines with this oxaloacetate and creates citrate. Citrate is a 6-carbon molecule. Then what happens is that it goes all the way around and all of these reactions occur, and we get more hydrogen ions here and here. We get some actual ATP here. We get some more hydrogen ions here, and lastly over here. So the process, what's happening here is we go from a 6-carbon to a 5-carbon to a 4-carbon, and then all of these other things happen, and we end up with oxaloacetate. This is important because if you don't have enough oxaloacetate, no matter how much acetyl-CoA you have, this can't run. This can't cycle. So this is a limiting factor. If you don't have enough oxaloacetate, and usually when you're starving or fasting, that might happen, you don't make energy. So this is one picture of the TCA cycle. I have a whole bunch in this video. Here's another one. And I basically just went through all of that. We go from a 6 to a 5 to a 4. These are all 4s here. We end with this 4. It combines with acetyl-CoA, and we just keep going around. As long as we have oxaloacetate, we can make more energy. And yet another diagram. So one glucose molecule through glycolysis yields 2 pyruvate molecules, and they, as they get transferred into the mitochondria, lose some carbon and add some CoA, coenzyme A, and so we get these 2 acetyl-CoA molecules, which then enter the TCA cycle and create some ATP, 2 ATP, and some of these hydrogen ion molecules. So we get 2 ATP molecules, 10 NADH molecules, and 2 FADH molecules with the turn of the cycle. These are going to become important because we can use these hydrogen ions to make more energy in the electron transport chain. So we were here, right? We had acetyl-CoA, and we used it in the TCA cycle. It combines with oxaloacetate to make citrate, and it produces 2 ATP and a whole bunch of these hydrogen ion substances that we're going to then move to the electron transport chain. So the electron transport chain is happening in these little folds here of the mitochondria. And basically what's happening is that these hydrogen ions kind of get passed back and forth through this membrane, and it creates ATP. I'm not going to go into exactly how this happens, but these high energy electrons are passed, through this membrane, and it allows the body to harness some of that as high energy compounds, these ATPs. The approximate energy creation, and we say approximate because it depends on a whole lot of things. Every body is different, depends on what you may have eaten, depends on what kind of state you're in, whether it's a fed versus fasting state. Lots of things can change, but we do get 2 ATP from glycolysis and 2 from the Krebs cycle or TCA cycle, and we could get 32 ATP from the electron transport chain. Some books or some textbooks or some scientists believe that it could be 28. Some say it's as high as 34. Again, it depends. So we're using 32 as our kind of perfect environment energy creation. So this is my version of the overview of macronutrient metabolism. We have carbs, lipids, and proteins, which turn into glucose, fatty acids, and amino acids. All of these can be turned into some sort of small carbon chains, whether it's a pyruvate, which is a 3-carbon molecule, or an acetyl-CoA, which is a 2-carbon molecule. Acetyl-CoA can be dropped into the TCA cycle, and we get more ATP, but more importantly, we get these hydrogen ions that we can shuffle off to the electron transport chain, and from there, we get a whole lot more ATP. And here's yet another one, and this one kind of shows you a little bit more than the other diagrams we've seen thus far, but this is the pathway we just took. From carbohydrates, we turn them into pyruvic acid or pyruvate. From pyruvate to acetyl-CoA. Acetyl-CoA gets dropped into the TCA cycle. All of those hydrogen ions get used in the electron transport chain to make more energy. So just a quick review. All of your energy-yielding nutrients, your macronutrients, your carbs, your proteins, and your fat, can be broken down into acetyl-CoA. Acetyl-CoA can enter the Krebs cycle, the TCA cycle, or it can become fat. If your body doesn't need any more energy, it's going to turn that acetyl-CoA into fat. And many of the reactions release hydrogen atoms. So even in when we turn pyruvate to acetyl-CoA, we release some of those hydrogen atoms, and then again more in the TCA cycle, and those hydrogen atoms with their electrons are carried to the electron transport chain, where more energy in the form of ATP is harnessed. And ATP, also known as adenosine triphosphate, is that high energy compound that captures the energy from this process.